My name is Mark Reynolds, and I'm a faculty member at the St. Joseph's University in Philadelphia. Today, I'm going to tell you about our studies with FixL, an oxygen-sensing heme protein from Rhizobia melati, and the role of a conserved salt bridge in that oxygen-sensing mechanism. The FixL, this a recent crystal structure, was discovered in the early 90s. Under high oxygen conditions, the oxygen binds to the heme domain and inactivates the kinase domain, as shown here. Under hypoxic conditions, the oxygen can dissociate from the heme domain, and this is communicated to the kinase domain, which is activated. A histidine is phosphorylated with ATP. Then the response regulator FixJ can bind, it's phosphorylated, and then its effector can bind to DNA and upregulate nitrogen fixation. So we wanted to look at this mechanism in more detail, and so we designed site-directed mutants of two conserved arginines in the heme domain. One arginine, arginine 206 or arginine 200 in SM-FixL, can interact with the heme propionate when oxygen, or in this case cyanide's bound, and the other arginine can interact with oxygen or cyanide, just like similar to the distal histidine in hemoglobin. So we changed this conserved arginine that interacts with the heme6 propionate to a histidine, a glutamine, a negatively charged glutamate, and an alanine that removes that charge. And we got stable proteins. We also changed the arginine to an isoleucine, a hydrophobic group, and it doesn't produce stable proteins. So we did some initial UV vis and other spectroscopic studies, but we really wanted to understand the structural changes in our protein. And to do that, we collaborated with Reynolds1 at the University of South Florida using a technique called photoacoustic calorimetry. Photoacoustic calorimetry uses a laser to photolize off the CO, and you get resulting photoacoustic waves that you can measure. Okay, here you can see for FixL, you see an amplitude shift from the reference to your sample, and that and other information and the time information can tell you about intermediates that are being formed in your sample. So if you graph the energy in that amplitude versus the temperature dependence, you can get a plot for each phase that you have, each photoacoustic wave. And you see for FixL, we have two slow phases and a longer, or two fast phases and a longer third slow phase. Okay, from the slope, you can get the change in volume, and from the y-intercept, you can get the change in enthalpy. So if you compare these three phases to other known heme proteins, you can see that FixL, what's unique is we see three different phases, three different intermediates, okay? A model compound, a model porphyrin, you just see one. That first phase is the first few nanoseconds when CO is photolysed off. It's just going into the heme pocket. Okay, if we compare our protein to sperm whale myoglobin or BJ FixL, you can see those two phases. There's two phases there as well. We see an additional third phase. So the second phase, you can see we see a volume contraction and a slightly negative enthalpy change, which is more similar actually to sperm whale myoglobin than just to the heme domain of BJ FixL. So our protein has the kinase domain, and we think the second phase has to do with changes in the heme domain as CO leaves the pocket. And the third phase, the third intermediate, is due to the proposed conformational change in the linker and kinase domain that activates the kinase domain. Okay, so if we look at our mutants, if we change that arginine to glutamine or alanine, we see dramatic changes in the second and third phases. And this is similar to what you see with sperm whale myoglobin. And their change, if you change sperm whale myoglobin to glutamine or glycine, you remove that salt bridge, right? They propose that's involved in opening up the heme pocket in myoglobin, and so if you change that charge, you're going to get changes in the volume and the enthalpy, and they saw that, and we see that in FixL. So we think the second phase is this heme domain structural changes, and then the third phase is where it's propagated to the kinase domain, and that's a structural change in the linker and kinase domain. And so with that, I'd like to thank my students and Randy Larson as his students, and also a variety of funding agencies, and for you for your attention. Thanks.